In G-protein communication, a signaling molecule binds to a G-protein-coupled receptor. This causes a molecule of GTP to displace the already present GDP on the G-protein. The extra phosphate of the GTP energizes the G-protein, which causes it to find an enzyme to give the energy to. When the G-protein binds to an enzyme, the extra energy from the GTP activates the enzyme and triggers the cellular response shown by the white dot below. In tyrosine kinase communication, when two ligands, shown in red, bind to the active sites of two receptor tyrosine kinases, the two will combine to form what is called a dimer. When the dimer is formed, a phosphate will be added to each tyrosine on the dimer, shown by the bumps along the outside edges. In turn, each phosphate can activate its own enzyme, triggering a different cellular response, up to 6 per dimer. This is useful when several cellular responses need to be made, such as when we're dealing with fight or flight as well as adrenaline and other things. In a normal person, neurons send signals by displacing negative ions in the axon, or the sending part of the neuron, with positive ions. This process is facilitated by the myelin sheath, shown in yellow, which speeds the signal up so it's actually useful to us and can reach our muscles in time to be effective. The electrical signal is then converted into a chemical messenger called a neurotransmitter at the end of the axon, which then binds to the dendrite, or receiving part of another neuron, to continue the process until it reaches its target, generally a muscle cell. However, in multiple sclerosis, the body's white blood cells attack the myelin sheath and will even take chunks out of the neuron itself. This slows and will eventually stop nervous communication, immobilizing its victims because electrical signals can never reach the muscle cells. Thank you for watching our video.